Thank you for the wonderful introduction, and I want to thank Summer Sessions for inviting me out tonight, and thank you all for coming in from this beautiful summer evening / apocalypse, depending on which way you're looking. It's interesting how there's so much smoke in one direction and not the other. So thanks for coming out tonight, and I want to share with you some of my research tonight and also some of the partnership work that I'm doing with the state and also with industry and with communities in the state of Alaska, and some of the great work done by my students, some of whom are here today. As well. And we're going to talk tonight about cleaning up contaminants using native plants and their naturally occurring microorganisms that are associated with their root system. We'll talk a little bit about why we might want a technology like this, how it works, and also go through some examples of where it's been tested in Alaska, including a study that we're doing here at UAF together. And so I first want to share appreciation for all of the students and staff and partners and funders that have contributed to the work that I'll share with you tonight. I'll share a long-term study that's included lots and lots of different students, staff, and mentees from UAF, including some are in the room today. Can you raise your hands? Ben and Amanda here. We work very closely with the Department of Environmental Conservation. I want to thank Jim Fish especially for helping create opportunities for phytoremediation work, and Robert Burgess, who's a former UAF student. Arctic Data Services is a small, a wonderful environmental consulting company started by Rodney Guritz, also a former UAF advisee, and Hannah recently graduated from here and now works for him, doing phytoremediation and other things. And the community of Caltag, where our long-term study is based, who've been wonderful partners. We have good friends there who have been very supportive and helpful. Great labor crews, wonderful school, especially the kids and the teachers, we love them, and the community members. And Shannon and Wilson, and also our partners in the Czech Republic who send grad students over, and they work on our sites too, and it's been a great collaboration across all these fronts, and thanks to our many funders as well. So contaminated sites in Alaska, they're a big problem. We know that Alaska's beautiful and pristine in many places, but unfortunately, look at this map. This is the Department of Environmental Conservation map of contaminated sites. There are hundreds of them all over the state, in part due to our military history and industrial practices, and also just the fact that we use and transport petroleum for things like home heating oil and all sorts of things. And so these are all sorts of different contaminated sites. Some of the common contaminants are highlighted up here. I'm going to focus today mostly on petroleum hydrocarbons, sometimes abbreviated PHCs, so anything from fuel to crude oil. We'll focus on diesel tonight especially. One of the additionally sad facts about our contaminated site problem is that rural Alaskans are disproportionately affected by this. There are more contaminated sites per capita in rural communities, and they're less likely to be cleaned up. We estimate about 5 times less likely, and there are many reasons for this, including just the logistical challenges of cleaning up things, sites off of the road system, which raises the question of how do we clean up petroleum contaminated soils? Well, oftentimes the first step is dig and haul. You get big heavy machinery in there, dig it up and move it somewhere else with a truck or a barge or maybe a train, and you move it to something like a landfill or a treatment facility. So sometimes there are specialized landfills where you can adapt a landfill to accommodate contaminated soil, or use thermal remediation, which is basically heating soil to volatilize and collect the petroleum. It might be burned or otherwise treated after that. There are just a handful of these kind of facilities in Alaska, but as you can imagine, they're not located in every rural community. It's not practical to get things there. There are microbes, of course, out there who can biodegrade petroleum very happily with a little bit of help. And one of the strategies that's come into pretty common practice that takes advantage of microbes is bioremediation, specifically land farming, which is used in Alaska occasionally. And that's where you take the contaminated soil and spread it out and till it over and over, just like you're farming it, but with no plants. And what you're doing is farming the microbes. You're giving them oxygen, helping aerate the soil, and they can biodegrade that contaminant, that petroleum, more quickly. And also some of it will volatilize. It'll just kind of volatilize up into the air. So this is a really useful technology. It works quite well, but it has challenges. You need heavy equipment, which isn't always available in a rural community. You need routine, like regular labor. So all through the summer, somebody needs to go and till that maybe a couple of times a week. And that can be challenging. And also space to spread the soil. So that leads us to the question of, is there a better way? And what does better mean? Well, I mean cheaper, easier, less labor intensive, and ultimately more feasible in rural areas, so we can help to address this environmental justice issue of contaminated sites being untreated. And I think plants might be the answer. So here are some native plants of Alaska, some of our many different types of willows. And we've been doing some research on willows that I'll share with you. This technology is known as phytoremediation. PHYTO stands for plant, and remediation to remedy the situation using plants. So it's the use of plants to remove or detoxify or immobilize, to treat in different ways, different kinds of contaminants in the environment. And I'll talk a little bit about the different ways plants can do that in a minute. There are some pros and cons with phytoremediation. So it's cheap, or it can be very cheap. It's kind of a passive strategy. Once you get the plants established, they do it for you. You don't have to get out there and till every day. It restores vegetation at the same time. It's sustainable. It's got a low carbon footprint. It's actually pulling carbon out of the atmosphere. And sometimes it can even be done in place without dig and haul. Sometimes we still move it, the soil. And often there's a positive community perception of this approach, which is an advantage as well. But of course, cons, it can be slower than digging up the soil and burning it. And it can sometimes be tricky to implement well. You got to get the plants in place and happy and growing. But once you do, you can often just leave them alone and they keep going. So this technology has been researched in the lower 48 and around the world since about the 80s or 90s. And there are lots of different instances of phytoremediation being implemented very successfully in other places. One of the most common approaches is this strategy, phytoremediation using a hybrid poplar tree, which grow really fast and they have deep roots and they're really good at pulling water out of the ground. And so this is a case where they might be treating a contaminated groundwater situation. And we like to make the joke that it's a popular method because it uses populus genus. Thank you. But increasingly, there's been a focus on the use of native plants versus these hybrid plants. Especially in Alaska, you can imagine not every plant is going to grow happily here. We need to use native plants, and we certainly don't want to use invasive plants either. So this has been the focus of our work, as well as native plants. Let's do a little walk around the phytoremediation tree here and look at the whole suite of different types of phytoremediation technologies that are out there. It's not just one thing, it's a lot of different things. Ways that plants can help with contaminants. So one is something we call rhizodegradation, but it's where roots stimulate microbes to biodegrade organic contaminants like petroleum in the soil around the roots. So in this case, it actually destroys the contaminant in the ground using microbes that are just being helped by plants. But there are other approaches too, like phytodegradation. There's some contaminants that plants can pull up into their tissue and then the plant can biodegrade, not very many. This is a pretty uncommon strategy. Plants aren't very good at taking up petroleum in particular, too, so it's not great for petroleum, but it can help with a few things. Phytovolatilization, this is where, in the case of volatile organic compounds like solvents that might be in the groundwater, some plants like hybrid poplar can pull that up, and then it volatilizes out of the leaves, and so it moves it out of the groundwater into the air and gets rid of it that way. Also phytometabolism, this is one where plants can take up excess nutrients. Sometimes it's used at the outflow of a wastewater treatment plant where you have excess nitrogen or phosphorus, for instance, or a feedlot or something like that. Phytoextraction, this is where plants actually uptake and sometimes they can hyperaccumulate metals and even radionuclides. There was a famous case of the sunflowers at Chernobyl that can take up radionuclides into their tissue. This technology makes me nervous, I have to say, because you can imagine, what if something comes along and eats that plant, right? You could be exposing the food web to that contaminant, so it's not a strategy I like to use, but it's been heavily researched and can be helpful perhaps in some situations. Phytostabilization is where the plants just help keep the contaminants from moving around. So their roots might bind with them and sort of lock them into the ground. And phytohydraulics, this is where plants suck up and transpire or evaporate water out of their leaves, and this can help control groundwater plumes. So say you have a contaminated groundwater plume, if you pull the water out, it keeps that contaminant from migrating. So it's really just controlling the water. But plants, like especially hybrid poplar, can be great at this. But tonight I'm just really going to focus on rhizodegradation, again, where roots stimulate microbes to biodegrade petroleum in the soil. So we're destroying the contaminant, a contaminant that doesn't really go up into the plant, per se. How does this work? Well, there are this is a really active area of research because we don't 100% know how it works, but there's a lot of evidence that it does. And we have some evidence pointing to a few different mechanisms. And one is there's a longstanding understanding that plant roots just stimulate microbial activity in their vicinity. Roots are kind of leaky. They leak nutrients and sugars and things like that, and more microbes grow. There's different microbes around the roots also than in the bulk soil. And so it could just be a general rhizosphere, we call it stimulation effect. But also plants release other sorts of chemicals too. Like plants produce chemicals that sometimes prevent animals from wanting to eat them. So things like flavonoids and phenolics, the things that like make a pine smell piney, the things that make a snowshoe hare not want to eat a spruce tree or certain willows. Plants produce lots of compounds for lots of reasons, including defense and pigmentation. So there are lots of plant compounds. They have chemical structures like this that are aromatic or even aliphatic, and they look a lot like contaminants. They're very similar chemically, actually. So there's also been a hypothesis that has emerged that plants that have this kind of chemistry might release those chemicals into the ground and encourage microbes to grow there that eat those kind of chemicals. And then that makes those microbes more readily able to biodegrade contaminants as well. So they're sort of enriching and stimulating them. So this is one hypothesis behind this. And one example that's quite well known is that of salicylic acid, also known as salicylate. It's basically aspirin. It's common in willows in particular. And it's one reason that there's a traditional medicine to chew on willow bark when you have a headache, right? It has an aspirin. It's aspirin, and it has an analgesic effect. But this is common in willow plants, and it actually turns on genes in bacteria for the biodegradation of naphthalene, which is a petroleum hydrocarbon, one of the toxic ones that's aromatic. And it just turns those genes on if they have them already. So it's one way they can flip the switch and get microbes to degrade contaminants more. So for this reason, people have studied willows a fair amount in phytoremediation, including us here in Alaska, where we have willows all over the place. You probably have them in your yard. They can be a little pesky sometimes, right, to get rid of. I'm cutting them back every year and they keep regrowing. There's lots of different species here. We picked one a few years ago just to start with, and it's called Salix alexensis, Alaska felt leaf willow. It has nice furry undersides of their leaves. You might recognize this one. And so we started testing this. Kelly McFarland, when she was a grad student, did a pot study where she grew some Salix alexensis in diesel-contaminated soil just for one summer, with and without fertilizer, with and without willows, and looked for effects on diesel degradation. And what she found is in only 3 months, there was greater diesel loss in the pots that had the willows compared to the unplanted ones, with or without fertilizer. And also when we looked at the microbes in that soil, the pots that had willows actually had more microbes that degraded diesel in the soil, which was exciting. So maybe they're enriching the diesel degraders. And then when we looked at which microbes there were, we could see that where there was willow and fertilizer, that actually shifted the microbial community, including the ones that were actively degrading naphthalene, that two-ringed compound I showed you before. So there's some reason to think that Salix alexensis might be having this effect in the root zone and could be a promising phytoremediation species. So hold that thought. As we address this question, well, has phytoremediation been tried in Alaska? So that was a lab or greenhouse study. Has anyone actually tried it out in the environment? The answer is yes. Yeah, including some DEC members who are in the back too. Yes, and I want to showcase a few examples. And this was actually started in Alaska, to my knowledge, by the Army Corps of Engineers, Cold Regions Research and Engineering Lab in the 90s. So they've done a series of different experiments throughout Alaska back then, including this one, which is on Farmers Loop Road in the Krell research area. It's kind of near the Steese Highway, back behind the fence. What they did is to bring in this is diesel contaminated soil and then crude oil contaminated gravel pad material, and divided it into plots and did different treatments where they fertilized and they added grass seed to some, did both in some, did nothing, tried all the different treatments and then monitored it. And what they found is fertilization and planting with grasses accelerated diesel degradation and crude oil degradation in the first 2 years. And then, as sometimes happens with research studies, they moved on to something else because, funding cycles change. And so that site was effectively abandoned. And then Jim Fish at DEC, I am so grateful to him for reaching out to me back in about 2011, and he said, hey, there's this old site, you might want to take a look at what plants are there now and how far the degradation has progressed. So that's what we did with several students in my lab. Starting in 2011, we went and did a follow-up study, and you can see the site has been completely vegetated by native local plants who were just they ran free, moved in, volunteered to grow in this site. And so we went back and cataloged the plants and the contaminants, and we found that plants, the original treatments of seeding and adding fertilizer, that changed the trajectory of the plant community as it entered the site and developed. So they went on different successional trajectories. And each of the different plots had different microbial communities associated with it. So the plants were probably affecting the microbes. And also, we looked at the diesel degradation, and we found a correlation between the plots that had more woody native plants, including willows, and lower levels of petroleum. So there's an association. We thought possibly there's a cause-effect relationship here. Maybe the plants accelerated that. So with all this results from these two studies combined, we thought what we really need to do is a controlled long-term study where we can watch it the whole time, where we might start with grass to get roots going, get plants established, get that rapid degradation beginning, as the woody plants could start to grow and develop more root systems, deeper root systems like willows. So what if we tried planting grasses and willows, maybe tested fertilizer? So we were hoping that maybe we'd get lucky and find the perfect site to do this. And again, thanks to DEC, Department of Environmental Conservation, we were introduced to this site in Caltag, the community of Caltag. Their traditional name means the camp for king salmon fishing. It's on the Yukon River in western Alaska. And it's one of many sites that has, unfortunately, schoolyards heavily contaminated with diesel from aging and leaking heating oil storage tanks that were put in when the Bureau of Indian Affairs built the schools in the mid-20th century. So those tanks might leak or rust out over time, and so there are these unfortunately contaminated areas right where you don't want it, where the kids are playing every day. So this is just a site we wanted to test phytoremediation in, and then hopefully if it worked, maybe that would open the door to implementing it in other schoolyards and other communities where there are these kinds of issues. So this is the aerial view of the school area. This is the site assessment by Shannon and Wilson, an environmental consulting company that the state hired to characterize the site, and the school building here. And all the red zones are where there were diesel contaminated areas. So it's all around the school, all around teacher housing, including, it was so bad near teacher housing that in the spring when the snow melted, a sheen of petroleum would come to the surface and then run downhill into town. So it was a real problem and really needed to be addressed. So the way that it was addressed in this case was to dig up that soil and move it to the edge of town, because we didn't want it to stay in the schoolyard any longer and wait for it to be cleaned up. So this was a case where dig and haul locally made sense, to dig it up from near the schoolyard here at A, and then move it out to an old gravel pit area in the community where there's just less foot traffic, where we could start to treat it and do this experiment. So the soil was spread out about 18 inches deep here in the phytoremediation experimental plot, and then a land farm was put in right next to it so we could do a comparative study and see which went faster and how things progressed. And here we are in 2014 setting up the phytoremediation site and the land farm. Here's EPA coordinator of Caltag, Doreen Nicolai, helping us out with student at the time, Rodney Guritz, here, who now is the owner of Arctic Data Services, and Jim Fish from DEC here with the land farm. So laying out the site, the initial diesel concentration was about 2,200 ppm, milligrams per kilogram of soil. And then we divided up that plot into 40 plots to do our experiment. And this is, if you were looking down at the plot, we planted some squares, the green ones, with willows, and some with a grass, a local grass that we found was used often in Alaska for revegetation, like stream bank restoration or other revegetation projects. It's a native grass species that we were able to get seeds for from, I think it was DNR. and we knew would do well in that region. And then we tried both together, and then plots with no plants planted just to see what moved in on its own. And then half of the site we fertilized, and the other half we didn't, just a one-time fertilization to see if that helped. And then we monitored this over, we're getting to be about 8 years now. And then it was time to plant plants, and we also wanted to continue doing community engagement throughout this whole process. So we did a spring break science camp actually in March of 2015, where we worked with kids on all sorts of topics related to plants and willows and microbes and proper disposal of oil and things like that. We had a lot of fun with the kids, and they helped us to take willow cuttings locally near the gravel pit, or near the school actually is where we are here. We had a boys and a girls team to see who could cut the most in 30 minutes, and the boys won. But we have some super cute members on the girl team too. So the idea here is, one of the wonderful things with about willows is you can cut them when they're dormant like this, and then plant them directly in the soil, and they send out roots and leaves. You don't have to start from a seed. So they're easy to propagate, but you need to cut them when they're dormant. So, it was March, so we needed to store them. So Rodney came up with this ingenious idea that we could build a field cooler by digging a hole in the snow and using extra straw that was left over from the Iditarod. So Caltag is a stopover on the Iditarod, and they always have extra bales of straw. So we layered straw and willow cuttings and snow and piled it all up, and it really did a great job from March all the way till June, keeping the willows dormant. And then we came back in June 2015 and planted the willows. Here's Ben out here watering some new willow propagules. And Rodney and our 2015 awesome team of local laborers from the city of Caltag who were helping out. So we planted all these different treatments, divided up our plots, and then, of course, allowed volunteer plants to move in too, which they're going to do whether we want them to or not. And we monitored this. So every year, except during COVID years, we went out every end of the summer and did soil sampling. So we used a soil auger and drilled and collected soil samples from top 9 inches and then 9 to 18 inches below that. That's the depth of the contaminated soil. Here's Amanda augering last year. And we collected those samples to analyze diesel and also microbes. And then we did plant community analysis by looking at all the plants that were there and what their proportion of abundance was, and measuring willow growth too. So here's a little time course of how things looked. So after the end of year 1, so August 2016, this half is the fertilized half of the site, and this is the unfertilized. And the fertilized ones, you can see how much bigger the grasses are. They've grown all the way to maturity and gone to seed. The unfertilized ones, they're eking along. They're just getting started. And you can see a lot of bare soil there too, but they're getting started. So definitely fertilizer helped the plants establish rapidly. And there was significant diesel loss in all of the fertilized treatments, especially in those shallow soils that first year, but not so much in the unfertilized one. So we can see fertilizer and seeding and planting was already having an effect in year 1. 2016 was a really rainy summer, so this was an interesting lesson as well in the comparison between land farming and phytoremediation. That this is the land farm, it was a mud pit, and they had to stop tilling it because the equipment would get stuck in the mud. So treatment stopped in the land farm for a stretch of time that summer, but the willow plant's happy. Willows love wet soils. They're doing just fine. No problems there. So year 2, 2017, you can see actually the plants are really filling in throughout the whole site. The unfertilized are starting to catch up a little bit with ground cover. Moving in, here's year 3. You can see the willows getting tall. We're doing some plant analyses. Kristen Hruska and Ben Hedges again here doing some transects, looking at the vegetation. And year 4, boy, those willows are really getting tall. Here's Ben and undergrad Kyle Caligari monitoring. See also how tall those grasses are. And we took a little pause for a few years in between there. Due to COVID, we didn't want to go out to the village and bring it in with us, so we came back in 2022. Here's year 7. Actually, the state sent Shannon and Wilson out to do just some limited sampling, and boy, the willows got really tall. They're getting thick here, thanks to Drew Frick for going out and collecting these samples. And just pulled a few samples from here and there to get a sense of what was happening. And all 5 of the shallow samples from 0 to 9-inch had met DEC cleanup limits, which means the levels were below 250 parts per million. But a few, like many of the deep samples, exceeded limits, so we knew that the deeper soils needed more time. Deeper soils have less oxygen. It's probably taking more time for the roots to penetrate as well. So it's not unexpected that deeper soils need a little more time. Here's year 8. So we went out last summer with new PhD student Amanda Stromecki, who is outstanding in her field. Get it? Okay. That's Amanda back there. Okay, and so we did, along with Shannon and Wilson, some quick field screening, and we're still analyzing the data on the exact levels of the contaminant for this year and all the previous years. This is one of Amanda's big jobs as a PhD student, is to put it all together. So we only have preliminary results at this time. But year 8, with a quick screening, we could see that all the shallow soils had met limits, cleanup limits. So the surface soils were clean, but some of the deeper soils still exceeded it. But it's only 7 out of 40 of the samples we took, so it seems like it's getting cleaner. It's catching up to being closer to clean, so this is exciting. Undergrad Kyle Caligari, who I pointed out earlier, he did an awesome senior capstone project for his biology degree where he looked at all the vegetation from 2016, 17, 18, and 19. And this is too much information for me to process, I know that, but I just want to point out, so each of these is a percent cover by different plants in that plot, or that treatment plot. So here's a control fertilized plot. And the green bars are the grasses, and the magenta ones down here are willows. So you can see here's a plot where we planted grass and willow, and they make up most of the composition of that plot in the fertilized half. But in the unfertilized here, we have a lot of bare soil, which I showed you earlier, right? But still some grasses and willows are eking along. And as time progressed, what you see is the willows get bigger, the grasses fill in, and there's different kinds of grasses. So the grass, it turned out the grass that we planted eventually was overcome by local grasses that moved in that seemed to do better at the site. And they're just as big and thick growing grasses. So there's a greater diversity of grasses, not the one we planted, even though it's native, but still lots of ground cover and lots of other herbs and forbs and legumes and things moving in as volunteers. And the willows are getting bigger and bigger, these pink bars where we have willow plots, until we have complete plot coverage with really diverse plant communities, including this aqua one, which is horsetail. You get it in your garden a lot. So this is kind of like what your garden would look like if you stopped doing anything to it. Very diverse. So anyway, what we learned from this is definitely fertilizer aided the initial establishment of plants, and native local plants really rapidly moved in and colonized and increased plant diversity and coverage. And the grasses we planted were replaced by local grasses, and the willows did great. They're continuing to thrive, and they're getting really tall and big now. So what happened in terms of diesel degradation? Again, we just have preliminary data, but I'll give you some teasers from Amanda's data analyses. So far, what she's done is to look at the diesel decay rate in the first 4 years combined. So that means a larger value means a higher degradation rate, which is good. So the fertilized plots, if you lumped them all together, they're degrading faster. The diesel's degrading faster than the unfertilized ones. Fertilizer helps, and this is something we've seen before in the literature, and it's being reinforced again here. Fertilizer helps. What if we break it out more into the different treatments? So here's just the fertilized plots and the 4-year diesel decay rate, again preliminary. Interestingly, the unplanted plots are doing great. Although notice that the spread is pretty large. So we have all these different points in here. There's a lot of heterogeneity in terms of the amount of diesel, and that's partly because it's a little bit patchy when we laid it out. And also the deeper soils are included here. They're still a little bit more contaminated at this point than the shallow soils. But if we look at the medians, the unplanted is doing pretty well compared to grass and willow and grass, but they're all moving forward. This raises an interesting question as to whether maybe the local vegetation might be doing better than the ones that we deliberately planted. And there's, we just picked 2 plants to try, right? Felt leaf willow and this Arctograssus latifolia, but there could be other plants out there that are even better. And we hope this study with good statistical analyses might actually reveal that to us, which plants that moved in correlated to greater degradation rates. Could be something else that we haven't thought of. But still, active planting is probably a good idea because it helps get cleanup started sooner, gets roots established. This is an overview of all the data. We just looked at these fertilized different treatments here. Here's the unfertilized ones. You can see how all the different treatments, they tend to still have a lower rate, but they're still degrading. And here's the land farm here in the middle. This is kind of interesting. It's very tight like that, and that's because it's being constantly mixed. We don't have the deeper soils that are degrading slow with the surface ones that are degrading less. And the overall rates are kind of comparable in some ways. But yeah, I talked about the depth issue. So is phyto just as good as land farming? Well, not exactly. Like the rates are kind of roughly similar, but the land farm actually reached DEC cleanup limits in 6 years. So it was clean in 6 years. The phyto plot, not yet, in part because of those deeper soils that weren't clean yet, which suggests maybe we should just not spread the soil so deeply. That might help it degrade faster when that's feasible. Or if you've got time and can wait, the phyto site seems to be approaching clean in 8 years, so it's not too far behind the land farm. If time is taking a little more time is okay, if that makes sense. What are the microbes doing? Well, this is another thing we're working on now, so I don't have a great answer for you, but except that everyone's working really hard on this. Here's Amanda again, who's a INBRI graduate research associate. Right now, and then we also had a partnership with Hutchinson High School teacher Robert Marcotte, who got funding from the Murdoch Charitable Trust to work in our lab in the summer and learn some new techniques, and he did a lot of DNA extraction from the soil. And undergrad Aria Workman and Amanda and some visiting students from Prague have been working hard extracting microbial DNA out of the soil so we can see which microbes are there and how many of each kind there are and how they relate to the different vegetation types. So what we do know, though, from some preliminary data, is that the land farm microbial community is very different from the planted areas. So that suggests, too, that the plants are really shifting the microbial community. So stay tuned for how and which plants shift it more in favor of petroleum degraders. We can't wait to find out either. We also had some fun with students just a couple of months ago in Caltag looking at microbes, too, in the K4 classroom with Amanda, leading some really cool activities with them. Okay, so summary of what we've learned so far from this site. Fertilizer helped. So fertilizer, oftentimes plants and microbes are nitrogen limited or phosphorus limited, so that can really get them kick-started. It increased plant growth and success, enhanced diesel loss, and this is consistent with other studies. The grasses, well, the one that we planted didn't persist, even though it's a native species, but other native local grasses moved right in and took over. And the plants shifted the soil microbial community compared to the initial soil and the land farm soils. In terms of treatment time and depth, so again, the land farm achieved cleanup in 6 years. The phytoplot is getting close in 7 to 8 years, and it's that 9 to 18 inch depth level that's really lagging a little bit behind. So those are some of our take-home messages so far, but stay tuned for more lessons from this study as we catch up with all the massive data sets. So now I wanted to kind of broaden it a little and just look at some other cases where phyto's been tested. But I thought, with actually help from my collaborators, like the small companies, the environmental consulting companies who are doing phytoremediation, and also the state, DEC, we compiled some examples of phytoremediation that's been tried in Alaska. There haven't been that many cases yet, so I wanted to share the ones we know about. I already mentioned the Corps of Engineers. They were really at the forefront of this in Alaska and other northern regions. They had what they called really demonstrations. They weren't really long-term experiments, but they were doing some research in Alaska and also in Korea in military contaminated sites. Here are some sites across Alaska that were petroleum contaminated, and they planted grasses and legumes and forbs in contaminated soils, and they also concluded that grasses really helped get vegetation established quickly, and so planting was helpful. But of course, as you've seen, eventually the volunteer vegetation moves in and takes over. But those weren't really long-term studies, but we could see some potential. Here's another study that Jim Fish from DEC shared with me. It's a site here in southeast Alaska, a former drum storage area that resulted in contaminants like gasoline, diesel, polyaromatic hydrocarbons and also chlorinated solvent contamination. So they are trying in situ phytoremediation, which means you don't dig it up and move it, you do it in place. And they planted native alder. So a rare case of alder being tried, and also Arctored fescue, which is a turf grass. And the alder didn't actually do that well, but the grass is doing great here. And they're going to do some soil sampling in the future and see how this is progressing. So we're excited to see how that goes. There's also one in town here that you can even see from the road if you drive around the back of the airport, the old Tesoro fuel depot. And this is one that Trihydro is a consulting company in charge of this, where with technology developed at the University of Washington, they're actually using poplar trees with endophytes. So these are bacteria that they cultured from the natural environment. They're not genetically modified, but they found bacteria that can degrade petroleum or degrade chlorinated solvents that live happily inside roots. And so if you dip the plant in a culture of these microbes, they'll colonize inside the roots, and they've found that it helps the plant survive and helps the contaminant degrade in some of their literature. So this is the first attempt of using this in Alaska. All this other research was done in the lower 48 with hybrid poplars or other poplars, but they actually used our local poplar species, balsam poplar, and these endophytes, and planted it in this old fuel depot in order to help do two things. One is to control the groundwater plume that's there, as I'd mentioned before, just get water out, but also to hopefully degrade some of the benzene and fuel and this dichloroethane. So this is also in progress, being monitored. They're reporting to DEC, and we'll find out how this goes in a few years. But the plants are growing after a little bit of a struggle at first to get them established. They seem to be doing well now. There's another one under development in uniloclete, where there's diesel, also polychlorinated biphenyls, trichloroethylene, and there's a plan to excavate soil and do phytoremediation there. So it's exciting to see people trying this out now, and the state seems to be approving these kinds of cleanup plans when they're well formulated. So this is going to be a good way to see how this works. But they have a plan to plant grasses and willows. And this project, they don't always go great. So this one was discontinued. It was a 4-year pilot study at Fort Wainwright. This was one that had a lot of different contaminants, though. It had gasoline, diesel, also pesticides, aldrin, dieldrin, and DDT in it. And so they said, they tested, what would happen if we planted, they planted willows, spike rush, hairgrass, and red fescue, and monitored it for 4 years. And then they saw, they were a little concerned about the results because some of the pesticides weren't breaking down. So then they pulled the plug on it and landfilled that soil and capped it. So that one didn't continue on, but it was a trial. So, but again, I think they were more concerned about the pesticides there. Here's another one in progress. This is by Arctic Data Services, which is Rodney Guritz, who again was a UAF student before. And this is a cool project in Ruby, in the village of Ruby, which isn't that far from Caltag. There was a former Head Start building, school building, that had also diesel contaminated soil. And so that was relocated for phytoremediation, and he used a very similar approach that we did in Caltag, willows and grass. And so far, what he found is the willows had a hard time, but he thinks that might have been a combination of too much diesel. There's really high levels of diesel, and that can be toxic to plants when it gets to be too much. And also, it was very dry, and so there's some evidence that drought can exacerbate that sort of contaminant stress on plants. So it could have been one or the other, or perhaps both there. But the grass is spread, and there's really high diesel loss, 91 to 97% in 6 years there. So he's also still monitoring that site. Here's a question you might be wondering: can you use this at your house? If you have a fuel spill, you or a friend, maybe asking for a friend. The answer is yes. Actually, so Rodney has a project like this in progress now in Esther at a private residence, where there was, unfortunately, a really bad situation where falling ice sheared a fuel line, and there was a big heating oil spill that went halfway into the crawl space under this house. And this is a really harrowing picture of excavating that. So, but his company worked with very limited resources with the homeowner budget to excavate that soil, and some of it was sent down for thermal remediation. But then some of it was just kept on site and spread out into the yard, and then they planted grasses and willow. And the grasses, grass and willow coverage were doing really well. The willows were surviving and thriving. And Rodney credits the homeowners who've been really great about watering regularly with the success of these plants. And so that's also being monitored, so stay tuned for results there. But this could be coming to a neighborhood near you, is phytoremediation. So what still needs to be done on the phytoremediation front in Alaska? Lots of things. Here are a few of my ideas of what I think is pretty important to address. More of these long-term studies on effective phytoremediation strategies for different regions and different situations. A better understanding of how plants and microbes work together, because if we really understand that relationship, maybe we can optimize it, right, and make things work even faster and more successfully. And this is an issue not just for phytoremediation, but across environmental remediation more broadly, is a need for more equitable and respectful partnerships with rural and indigenous communities in cleanup activities. So making sure that this is truly a partnership from the beginning. There's a long history of people sort of swooping in to clean up a site for a community, and while that's appreciated for sure, if the community is not involved and maybe they lose out on opportunities for jobs, of helping out with the cleanup, they don't maybe always have a say in the decision making, or they feel they might not. And so there's more we can do, and I know that the state's working hard on that, and that we're learning a lot more best practices in academia. And I know Rodney's really taught me a lot about this as well. Also to consider data sovereignty. So Amanda's been teaching me a lot about this as well, and the importance of making sure the communities know they own the data. These are their microbes, their plants, their land, and so we're not going to publish data without their permission, and they can determine what's done with the data or not done. And overall, just trying to help find ways to support communities becoming more environmentally self-determined and self-sufficient. So one way that we're also working together across industry and academia in the state is to prepare a guidance document for phytoremediation in Alaska. We were just meeting today to talk about this, that will hopefully help communities and remediation experts, so other regulators like DEC personnel, environmental consultants, communities, homeowners, understand what phytoremediation is, how it works, what are some best practices for putting that into place. So we're working on that and hope to get that out before long. Okay, with that I'd like to close and thank everybody one more time, and Basi in Koikana Athabaskan. Thank you.